Hey everyone, before I begin, I got a quick question. How many of you are dog owners? Show of hands. Great, we'll save that. My name is Carlos Munoz. I'm the CEO and co-founder of ReBlood RX. We provide an alternative oxygen delivery solution for a humane alternative. You might be asking yourself, a humane alternative for what? Well, it turns out there's a growing ethical concern of how blood is being sourced for man's best friend. Currently, dog blood is sourced through closed colony blood banking, which takes advantage of dogs bred and raised as living, breathing blood bags until their dying breath. This has forced California to pass legislation to end closed colony dog blood banking. As a result, this has left a huge gap in the market. When we're looking at how blood needs to be produced now, we're turning to the community of pet owners, where people like yourselves have to take your dog in to donate blood. Second question, how many of you guys actually take your dog in to donate blood? Not many of us. And that's the problem we're solving. ReBlood can provide a solution that is not only practical, affordable, and ethical, but allows us to bridge this gap. The way we do this is by taking expired red cells from humans that could no longer be transfused into patients, and we take a protein from them. And by doing so, we engineer it specifically so we can transfuse this and mimic red blood cells. This solution can now carry oxygen to any patient regardless of blood type and regardless of species, whether that be a human or a canine. And furthermore, it doesn't need to be refrigerated. It can be stable from minus 80 degrees Celsius all the way up to 50 degrees Celsius for 2 years. Now, we've been hard at work. We filed our patent. We have small animal testing showing safety and efficacy of the solution. And what we're doing now is we're moving into large animal testing. We are currently working with a manufacturing consultant in order to build a GMP facility. And we currently have a partnership with a large veterinarian hospital to run clinical trials. That same vet hospital is actually our first customer as well. And in order to accomplish this, we need money. $1.25 million in a seed round. And what this accomplishes is our first product to market by 2027. I'll repeat that. With a seed fund of $1.25 million, we can accomplish product to market. By 2031, we anticipate $150 million in revenue. And the way we're going to accomplish this is by taking 27% of the market share in the form of active veterinarian hospitals, about 48,000 of them in the United States. 27% of them will actually be carrying ReBlood. And from that, we can equate to $150 million year-to-year revenue by 2031. The platform technologies can be also leveraged in other forms, such as the DoD, EMS, anemic patients, as well as organ transplant solutions. This totals a $5 billion market. In order to accomplish such ambitious tasks, we need a strong core team. Myself, Ms. Daniela Lucas, and Mr. Christopher Caballero create the core team, and with years of experience from our advisory team, we can navigate these harsh waters. Thank you, everyone, and if you have any questions, please feel free to contact me.
Can you take me through again? You take human expired red blood cells, and then you do exactly what to it to extract the oxygen carrying capacity?
So we take out hemoglobin from the expired red cells, and we engineer it in order to become a transfusion solution.
Okay, so you take the hemoglobin itself, and
Correct.
now it becomes a protein, basically, that you're carrying in the fluid.
Correct.
Okay. And are there any potential allergic reactions that veterinary candidates can get by having this repeatedly?
No. As of now, from what I can tell from my small animal studies, in mice, rats, guinea pigs, and hamsters, there's no chronic immunological effects.
And so despite it being a allopenic, essentially, transfusion, even if you repeat it in the same animal, there's no allergic reaction?
Correct. So, I mean, if you'd care to know, I believe the reason being is because of native scavenger proteins that are universal in terms of haptoglobin and hemopexin. Further, if you'd like to jump into that, I'm happy to discuss that.
And then you also mentioned potential use in DoD, EMS, and other. You're talking about human usage
Correct.
of these. Where are you in terms of that development, in terms of regulatory approvals and the testing to be able to determine whether it's a safe use or not?
So for human market, in order to go down that pathway, we've been relying a lot on DoD. And thankfully, I do have a lot of support through the Air Force. And so with every dual-use technology, they do encourage a strong civilian use case. And so veterinary medicine is our strong civilian use case for beachhead. And so it's kind of going co-aligned. Truth be told, my conversations with CVM, Center of Veterinary Medicine, is the FDA branch, is much further along than my conversations with the FDA, specifically CBER for biologics.
Thanks for that presentation. This sounds wonderful. The question I have is, you mentioned that this would be bridging the gap from solutions that we currently have. So why don't you see it as a complete replacement?
The biggest reason is because it only has a short half-life of 21 hours in comparison to red cells, as we know have a longer. And so that's why it's really just meant to be a bridge or a gap in order to get to the point where you can actually transfuse blood, or at least limit the amount of blood transfusions that a patient would need.
It's along those lines. Obviously, you can saturate the fluid itself with increased oxygen pressure. How much additional oxygen carrying capacity is there by adding hemoglobin into the fluid itself?
So that also depends on, well, right now I concentrated at about 10 grams per deciliter, and it has a P50 of around 31, which is what we see in red cells. And so once you boil it down to the microcirculation, it kind of allows for perfect mixing, so to speak. And because it's laying around in the plasma, in the cell-free layer, it's really just kind of taking advantage of different biophysical properties. And so going a little scientific here is that you'll actually allow that play on with the colloid osmotic pressure and the viscosity to regulate the blood flow and thus the oxygen distribution in the tissue.
And the release of oxygen is not changed by alteration of the protein at all?
Correct. It still follows Bohr's law. So it's still going to be dependent on, normal mechanisms.
Any storage this?
So if you're asking between minus 80 and 50 C, if I store it in that sense, or what do you mean?
Well, I mean with DoD and just have something to store, transport.
Ideally, the solution when we test it, it's at room temperature. I've brought it all the way up to 50 C to see, what happens. And the reason why I stopped at 50 C is denaturing of proteins, kind of when you're pushing higher temperatures, and that's obviously going to be bad. But no, in terms of the solution, it's suspended in a modified ringer solution. So it's hemoglobin suspended in ringers.
And so it's transported like you would a red blood cell pack.
Correct.
Essentially.
Correct.
Any potential to concentrate it and then dilute it and be able to use it in the more?
Yeah, absolutely. That's kind of how I produce it. When I initially purify it, I purify it to about 20 grams per deciliter, but the functionality of it is usually around 10 grams per deciliter. And I just basically suspend it in ringers some more to dilute it.
So how do you get the expired blood? Like, is there a time that, you can't use it anymore?
Yeah, so I guess I should define expired blood. Expired blood usually means roughly 40 days, depending on the solution that it's stored in. And so that's kind of dependent on a number of factors, but essentially after that amount of time, the proteins in the red cells will no longer be kind of, lack of a better word, compatible with a healthy human and then cause a transfusion reaction. And so at that point, the solution, as it ages, becomes more at risk for patients. And so at a certain point, the FDA said, okay, no more after this point. And so those are generally just discarded. Talking with, different blood banks, I leverage the fact that they have to throw them away. So at scale, it's still a kind of ongoing conversation, but the idea is that they would either sell it to me at a very, very, very discounted price, or B, they would actually just pay for me to take it up. So I haven't got through that strong commitment yet, but that's kind of the two schools of thoughts that we're we're playing with.
Just following up on that. So at scale, is this something that needs to be done at a central facility, or is it something that the blood banks could do themselves at their facility?
Ideally, what I'd imagine, especially for like pre-hospital care or things like that, you would probably want to partner with the blood banks and actually have another thing on their product catalog. But I do envision a central facility where ReBlood would basically give out to medical distributors or blood banks alike through contracting, and kind of keep a strict hold on the manufacturing, as that's really the secret sauce of how I purify it and engineer the functional protein.
Is your purification process enough? Essentially, this could be like equivalent of a pooled blood, right? So because you're taking multiple red cells packed and combining them together. Any potential risk of disease transmission from the pooled blood?
That's a good question. And that's been one of the big things that what I've been reading from just FDA and blood products is now ever since COVID with zoogenic diseases and carrying through transportation. That's one thing that the FDA is going to be big on is there any at risk of acquiring any type of diseases through transfer? And for that, I'm not sure, but that's something that the FDA is going to be really big on.
Hi, Carlos. Great presentation. I just want to make sure I have the technology, that I understand the concept correctly. So this is meant to, in humans, be a bridge to transfusion, not so much the actual, not to replace the blood banks and everything. But in dogs, is it also meant for the same use case, or is this kind of like the last step and the dogs or whatever animals are going to be okay after this blood?
It's really meant to be a combination therapy, right? Where you infuse hemoglobin solutions for, let's say, if the dog needs, just play with the numbers here, like 4 bags of ReBlood and they only have 1 bag of actual dog blood to transfuse, they would be able to supplement ReBlood in order to limit the fact that they don't have blood on hand. And so the biggest thing there is the fact that, as I mentioned, it's based on community blood banking now, which limits the supply of blood altogether and causes a giant blood shortage.
Would they need to ultimately get a full transfusion with dog blood afterwards, or is this just kind of, does this hold them off pretty well for a while? Ideally, this would be able to limit the amount of blood transfusions that they would need and the dog would be able to survive. It's also a case-by-case basis depending on how much blood loss and things like that. But you could theoretically sustain or resuscitate a patient and allow the dog to carry on, assuming, other assumptions there. But technically, yes, you could resuscitate a patient with just this. Thank you.
One question. Did you talk to biobanks already, and you are positive that there is not going to be an issue for donating or like getting this blood samples? Because usually biobanks are collected with particular consent forms for a particular reason and purpose, right? If it's not already integrated when they were doing the collection, how is that going to be working?
Yeah, we've actually thought about that and the need for a new waiver to necessarily say, hey, when you're filling out these questionnaires for people who are donating blood, that what's going to happen to your blood at the end of the day and kind of the ethical concerns there. We're aware of it. We don't have a good necessary answer to your question, so I apologize.
So it's hemoglobin. Is it universal then?
Yes. So the same stuff that I'm trying in my small animal is going to be the same stuff we commercialize in canines, which is the same stuff we anticipate commercializing in humans as well.
But blood type?
Blood type, it doesn't matter because blood typing is what the remembrance.
Thank you very much, Carlos.
Thank you for that.